Hello, thank you for coming. Today I will be presenting the initial stages of our work, a strontium baseline of Mozambique for reconstructing Stone Age dispersals. Over the last 10 to 15 years, isoscapes have become an important tool employed by archaeologists to understand past faunal and hominin mobility by comparing strontium ratios in bone and tooth enamel to known geological strontium ratios. And this is all part of a massive effort to create a global strontium baseline. Most notably, this recent strontium isoscape for all of sub-Saharan Africa, published by Wang et al. 2024, is an impressive example of this. However, I want to highlight that as massive as this study is, these are predicted values based on machine learning and random forest generation. And of the 800,000 square kilometers of Mozambique, these predictions are based solely on 11 samples, which include seven soil samples from the Wang et al. 2024 study and four baboon samples from Domini et al. 2020. And while broad isoscapes are, of course, valuable, this gives us the opportunity to produce a high-resolution localized map for Mozambique, filling these gaps. My main research question that I want to understand is how variation of local landscape use and faunal mobility affect human behavioral adaptations to environmental change. By developing a bioavailable strontium isoscape for Mozambique, we are able to understand migratory behaviors of fauna, especially as a response to environmental and climatic changes over time. Just briefly, there are four isotopes of strontium. There's 84, 86, 87, which is radiogenic, and 88. So strontium 87 and 86 ratios are valuable for archaeologists because strontium values in bone and tooth enamel are reflective of where on the landscape these fauna were eating their food sources from. So, in an archaeological context, we are interested in bioavailable strontium, which is, of course, the portion of strontium from the environment that can be taken in by living organisms, i.e., fauna and hominins, and deposited into bone and dental tissues, like tooth enamel. So, to briefly break this down, strontium occurs naturally in bedrock and in various layers of ground soil, which is then taken in through plant roots. And plants also absorb strontium from rainfall, sea spray, and aeolian dust. And then fauna come and consume plants in their environment, thus taking in the strontium and depositing it into their bone and tooth enamel. Seasonal migration can then across areas with distinct geologic differences can then be extrapolated from striated analysis of strontium in dental enamel. Or an average can be derived from sampling the length of the tooth from the crown to the enamel root junction. Strontium in the environment is variable depending on several factors, but as seen in the previous figure of strontium pathways, distinct isotopic signatures exist in the underlying bedrock. Weathering and erosion occur. There's also aeolian dust and fluvial impact from river systems, carrying sediments and depositing them into topsoil. Our research area is located along the Save River in central Mozambique, in the Sofala and Inhambane provinces, where you can see older weathered bedrock like that of the Zimbabwe Craton, where the tributaries of the Save River originate. They have higher strontium 87 ratios because the bedrock had more time to undergo radioactive beta decay. Younger quaternary sediments, like alluvial sands and eluvial floodplain sediments, have lower ratios of strontium 87 because they have undergone less radioactive decay. So older sediments have higher strontium 87 ratios, and younger sediments have lower strontium 87 ratios. During the 2024 field season, we sampled along the Save River, mostly in the Sofala province, which is just north of the river, at 21 locations, including two archaeological sites, Zimora and Chesingulane, which are represented on the map as the two stars. And in order to capture any variation in local geologic strontium, we sampled three plants of varying root length at every survey point, which includes grasses, shrubs, and mature trees. Ideally, we do try to capture the change in geology across the landscape, as well as targeting surveyable locations based on this higher resolution geologic map. Additionally, we surveyed areas that appeared to have a visible change in surface sediments while we were out surveying on foot. And this is also why we do have some survey locations that look grouped together, or even overlap. However, we avoided areas with clear anthropogenic disturbances like farming activity and major roadways. Once plants were collected and dried, I then processed the samples at the Brussels Bioarchaeology Lab at the Vrije Universiteit in Brussels, Belgium. I digested the samples in acid, isolated the strontium through column chemistry, and then I ran the analysis on a new plasma 3MC ICP-MS. So what did we get? Well, a whole lot of variation, more than we expected. However, this is still interesting because this broad variation in strontium values is likely because all these samples come from within a floodplain of the Save River. So samples within each location do actually cluster very well and have smaller variation within uh, survey locations. And this is really proof of concept, and that the isotopic variability we see between locations is likely due to a geologic influence. A few outliers at MS1, which was the most coastal location and directly on the river delta, we have a mean of 0.71017, which I firmly believe is heavily affected by sea spray. The mean modern marine strontium value is 0.7092, which means that it is less than one thousandth of a point different than our own average value. The only truly bizarre results were at SVS 7, where the grass had a significantly higher value than the tree sampled at the same location, and significantly higher than the rest of the samples at all of the survey locations. While there weren't any blatantly obvious disturbances like farming, there are two possible theories for why the sample is so different than the expected range. First, this sample includes grasses from two survey locations that were very close together and visually overlap on the map. The second is that there may be disturbances to the topsoil causing these results that aren't immediately visible, like influence from the alluvial sediments from the Save River. I am planning to return to Brussels to analyze material excavated during our 2025 field season, at which point I can rerun these grasses and try to figure out what is happening at this survey location, specifically with these samples. Here I mapped the bioavailable strontium values for each survey location, for each analyzed survey location against the geologic categories. And again, the resulting strontium values appear somewhat variable, even within these broad geologic units. We ran multiple regression analyses to understand what the driving factors of this isotopic variation are, including elevation, distance from the Save River, distance from the coast, and geology. And while geology is somewhat a driving factor, the lack of significance between the majority of the geologic units suggests that the alluvial and colluvial mixing is the biggest factor for the variation that we see, which does make sense being in a floodplain of the Save River. So how do we address this? Well, we just need to sample more locations. During the 2025 field season, we excavated at a site, Caverna do Coluche, which is a series of limestone caves near the village of Bushani, represented by the lowest star on this map. We targeted survey locations, represented by the green dots, in the surrounding region based on the high-resolution geologic maps to capture the geologic diversity and hopefully the strontium diversity of the region. As of right now, I am planning to return to Mozambique in October for additional fieldwork, at which point I plan to collect more plant samples between Gorongosa National Park and travel south towards Maputo. As I mentioned, isoscapes are an incredibly important tool for understanding past mobility and fauna across different geologic settings, which of course can then be used to further extrapolate human behavior. During the 2025 field season, we found a significant amount of material, including ostrich eggshell beads and fragments, and over 200 mammalian teeth. So future work will include strontium analysis of these materials, which will contribute to creating a baseline and help to reconstruct localized fauna mobility as well as human landscape use. I would like to thank the session organizers for inviting me to share this part of my dissertation research. Thank you to Doctors Christoph Snoek and Hannah James at the Brussels Bioarchaeology Lab. Thank you to my supervisor, Dr. Gina Buckley, and to the entire Dispersals team. Kanimambo, obrigada, and thank you.